I'm going to start out by illustrating basically what the problem is. Hopefully you'll be able to get the sense from these images, which unfortunately will suffer from the quality of the projector. But here's a lakeside scene in which you should be able to easily extract things like colors and boundaries and edges and construct for yourselves and extract from this image objects that you recognize, like this dome and a bell tower and some other things. Our visual system, of course, is designed to allow us to make sense of the visual world in this way. And here is the same scene rendered as though seen through the eyes of a newborn human infant. And despite the fact that the projector is poor, you should be able to get the impression that this is the same visual scene. And what it should convey is the newborn infant visual system is, in a way, undeveloped, it's immature, and the sensitivities, as I'll show you, are so poor that it precludes the ability to actually extract these boundaries and edges and make sense of the world. And the thing that my research is concerned with is how do you get from the infant image to the adult image? In other words, how does the visual system develop in such a way that it supports adult-level perception that allows us to make sense of the world? And I'll talk a little bit about, or a lot about, the psychophysics, things that change during development, and I'll talk about some of the limitations that we know about in the early visual system, and gloss over a lot of other ones. And then I'll talk mainly about global visual processing, what I mean by that, and how it may depend on processing in the extrastriate visual pathways. So first, I want to say a few words about the animal model. So the animal model I study is the macaque monkey. This is an image of an infant macaque. And you might say, well, why bother? We've got kids everywhere, right? And so the importance of having an animal model in this case is that you can study development under controlled conditions. You can study it longitudinally. Your subjects always show up for their experimental appointments. And you can importantly study the underlying neural mechanisms in the same subjects. And sometimes, in some cases, longitudinally as well. And so this gives you a very powerful tool for understanding the neural mechanisms underlying development. Now, why the macaque monkey as opposed to another animal? The visual system structure and function, as many of you know, is quite similar in macaques and humans. And I'll show you visual development across species is virtually identical. And so if you're studying development, it's important to know that the developmental process is similar. And then finally, importantly for my research, macaque monkeys develop many of the same natural visual disorders that occur in children. And so we can study those disorders as well and understand those neuromechanisms as well. So we're going to start out looking at the development of vision, just simple visual acuity. There are, as you'll see, many metrics of vision. This is the most basic. It's visual acuity, if you will, based on the eye chart on the right. Our metric for visual acuity here is grading acuity, shown on the left-hand ordinate. On the right-hand ordinate is the eye chart equivalent, with the big letters now at the bottom and the small letters at the top. And this is showing you acuity development as a function of age in weeks for a group of infant monkeys, some tested longitudinally and some tested cross-sectionally. And what you can see easily is that there's a nice coherent developmental process that asymptotes by the end of the first postnatal year. And so their acuity is adult-like by the time they reach the end of the first year. And how does that relate to human visual development? Well, I've just overlaid on that graph group data from human infants from other studies, and what it shows is there's an almost perfect correlation, especially in these ages when the monkey is developing quickly, between the macaque monkey and the human in terms of their acuity development. And the only trickery here is I've now plotted monkey age in weeks and human age in months. And so if you consider a 4-to-1 translation, monkeys develop approximately 4 times faster. The asymptotic level is similar across species as well. Now, acuity is only 1 point in terms of visual function. It tells us what the finest resolution is of the visual system. The high end of this grading pattern, if you could see it, would be out here. That would be acuity. And what this is supposed to represent is spatial scales below the acuity point or across the spectrum. In a sense, you can draw out the modulation transfer function for your own visual system. It's an inverted U-shaped function. As you go higher on this slide, contrast gets lower. In this case, contrast is low everywhere. But in any case, depending on where you draw your function, that describes your contrast sensitivity function, your sensitivity as a function of spatial scale, from coarse down here to fine up here. And so if you look at the development of contrast sensitivity, you find a parallel, if you will, between the development of the cutoff, that's equivalent to the resolution point, and the whole contrast sensitivity function. These are data, contrast sensitivity as a function of spatial frequency, like the demo slide, that are taken from an individual infant monkey as a function of age longitudinally. And his ages here are shown by the curves. And what you can see easily is that as acuity is improving, contrast sensitivity is also improving. And that entire curve does not change shape, it shifts up and to the right in that space. And so over here, I'm showing you as a function of age, peak contrast sensitivity, and spatial resolution now extracted from those curves from a whole bunch of infant monkeys. And you can see that those two aspects develop in concert, with sensitivity and scale developing similarly. Now, this is the typical spatial vision profile. This is a representation of temporal vision, basic temporal vision, and what this is showing you is temporal modulation sensitivity as a function of age, again now for an individual infant monkey tested at these ages. And you can see that this is also an inverted U-shaped function. It's not the same one, because now you're talking about a different domain of vision. However, it shifts up clearly with age, now asymptoting actually at an earlier point, and resolution on this, which is temporal resolution now, or you might think of it as a critical flicker fusion frequency, so they're analogous in a sense, is almost adult-like at the youngest age, whereas sensitivity improves with age. So unlike in terms of spatial vision, if you look at the change in the peak temporal frequency and the cutoff for these curves, they do not parallel one another. And so modulation resolution, temporal resolution, is very consistent across age, starting from the earliest ages that we measured behaviorally, which is 3 to 5 weeks. After birth, and going on up until asymptotic level, which, as I said, for temporal sensitivity is earlier than it is for spatial resolution. And those are two plotted together with spatial development on the left, with spatial development on the left and temporal on the right. And you can see that those two basic functions of vision develop differently, okay, over different time courses, where temporal resolution, temporal sensitivity develops faster and asymptotes earlier. And in the case of spatial vision, both the resolution and sensitivity develop in concert, and in temporal vision, that's not the case. And the point I'm trying to make here is not everything develops at the same time in terms of vision. There is no single developmental time course for vision. All right, and depending on the metric that you're looking at, you get a different profile, and that suggests that you're looking at different underlying neural mechanisms as well. What that means is that if you look at even different basic visual functions, here now I've compiled 4 different ones. You've already seen spatial contrast and resolution and temporal modulation sensitivity. I've added vernier acuity, which is a spatial positional sensitivity. And you can see that even in that case, there's a different developmental profile. And that suggests that, in fact, when you're talking about trying to understand the neural mechanisms underlying visual development, it's not the case that there's a single gate downstream that is maturing at some level to allow visual function to mature. Each one of these different functions develops based on different aspects of the visual system, and they have their own developmental time courses. Now we're going to look a little bit at some of the things that change and some of the things that don't in terms of the underlying neural processes. And this is just a bottom view of the brain, like that Semir showed, as drawn by Rakich. And there are many things that change about the organization of the retina and about sensitivities and the organization of the LGN, postnatally as well as prenatally. And we've done a lot of studies to look at whether or not those changes actually set a fundamental limit on the developmental processes that I just showed you behaviorally. And except for one case, which I will show you, the answer to that is no. So despite the fact that you have anatomical and physiological changes earlier in the visual pathways than the primary visual cortex, they seem not to set an important fundamental limit, or a gate, if you will, on the development of processing. So we'll look at a little bit at area V1 and how things develop in V1. And what I'm going to show you here is one of the first properties that's unique in the visual system when you get to the level of the visual cortex. As many of you know, it's the first site of convergence in the cortical pathways of information from the 2 eyes, binocularity. And as demonstrated beautifully by Hubel and Wiesel in many studies in animals, there's an organization called Ocular Dominance, which is a waxing and waning of the influence of the 2 eyes in the cortex, and that can be represented in a histogram of this kind, where all of the middle categories, 2 through 5, in this histogram are binocular at some level, and the categories 1 and 7 are purely monocular. And what you can see here is proportion of cells as a function, eye dominance, as a function of age. And so these are histograms from a number of animals taken at each age, recorded from 1 week up to adult. And what you can see is that the basic distributions in macaque monkeys of ocular dominance do not change with age. If we can look at a variety of other properties that are perhaps not unique to cortex, but certainly representative of cortical cells, both as Semir and Tony have also already told you, including direction selectivity and orientation tuning, plotted as a function of age. And the scales for each curve are shown here on the ordinate that are color-coded to them. And just for example, orientation and direction selectivity, you can easily see those properties do not change as a function of age. Recording as early as 1 week in infant monkeys, those properties largely do not change between then and adults. Now, there are some things that do change, and importantly, throughout cortex and also at the level of the LGN. If you look at basic responsiveness of neurons, this turns out to be important down the line. It improves quite dramatically between infants and adults. So infant neurons, while they're well-tuned, say, for orientation and direction, fire relatively few spikes. So they actually convey pretty good information, but they don't generate a lot of spikes to do that. As neurons mature in the brain, they generate many, many more spikes and send out much more information. Spatial resolution, importantly based on our acuity studies, behaviorally does change with development at the cortex. And I'll show you in a minute a direct comparison between the rate of change of spatial resolution of individual infant neurons and behavior. And so that's shown here, where now I'm showing you the same behavioral data in the small dots here, where we have spatial resolution plotted as a function of age in weeks. And what I've plotted on top of that is also resolution of some of the neural aspects of the brain. In red, this is the resolution based on anatomical studies of the infant retina at the different ages indicated. And in the colors, the LGN resolution and V1 resolution. And there's a couple of takeaways from this slide. The first is that none of the anatomical curves parallel the behavior one, okay? So the extent of development that we see behaviorally is about a factor of 30. The largest change we see in terms of any of the neural metrics that we've used is about a factor of 3. So there's a serious mismatch there. And it's also the case that the development of resolution at the level of the LGN and at the level of V1 largely simply parallel the periphery. So resolution as set in the retina essentially sets a limit on the neural resolution going forward in the visual system at these levels, but those seem not to actually set a limit on behavioral change. Now, the one thing that does seem to relate clearly between, or at least possibly, between neural elements and behavior is temporal resolution. So these are the temporal modulation sensitivity and resolution data that I showed you earlier. And plotted on top of those are resolution data from the level of V1, V2, and also the LGN. And you can see that the physiological curves overlay one another quite well at every level. But in terms of overall modulation sensitivity, like in terms of spatial vision, there's a mismatch. However, in the terms of resolution, it may well be that temporal resolution is set already, perhaps at the level of the retina, but certainly at the level of the LGN. So that's the one thing that sort of is against the rule that I mentioned earlier. So what have I told you so far? There's limitations on neural and spatial and temporal resolution. Spatial and temporal resolution that are set very early in the retina at the level of the LGN, but these seem not to have, at least in terms of spatial vision, any real restriction in terms of behavioral sensitivity. So there's a mismatch between the neural development and the behavioral development. And what that means, realistically, is that there's much better information available in the infant brain than the infant confesses to being able to use. And that information is not being used to drive behavior. And so the question is, why is that? So there are a number of possibilities for why that might be true. And one obvious one is that if the visual system development is a hierarchical process, and there are many downstream areas, as we've already seen, then information feeding forward may be limited further downstream, okay? And that information may be restricted, the motor output, and other decision processes. And so we're going to look at some of those downstream processes, and I'm going to add to this slide here that I showed you before, data on the left, from a variety of different basic visual processing, showing you a variety of profiles. Well, on the right, what I've added is what I'm calling global functions, and these are ones that are, in a sense, higher order. They build on lower level visual processes. They require the integration of information over space and time in order for you to solve these problems. And things like global motion sensitivity, and I'll show you some pattern motion sensitivity, and processes like what we're calling contour integration, I'll show you in a minute, where you're linking elements across space in order to extract a global form. And that's, of course, essential for being able to understand the forms that are out there in the visual world. And so we're going to look at a couple of these global visual processes, the one at the top, global motion, and at the bottom, we're going to talk about not contour integration, but another aspect of global form development, the one that Tony talked about. But first, let me just show you what I mean by contour integration to make sure you have an understanding for what I mean by global form and get a sense for how this develops in relation to acuity development. So this slide shows, if you can see it, a bunch of randomly arrayed little Gabor patches, these little grading patches, right? And they're all differently oriented and scattered across the screen. And there is embedded in this image a form. And you should be able to extract it if you have good contour integration ability. And that form is a circle. And what we challenged infant monkeys to do is to tell us where in this field of randomly arrayed Gabor patches this circle, the coherent circle of elements, exists. And for those who are struggling, either because your contour integration isn't great or because the image is poor, it's right here. And so this is a task that most adults, but not all, can do relatively easily. I don't even know if you can see the circle. Hopefully you can. But in any case, infant monkeys, it turns out, have a great deal of difficulty doing this. So here's the same acuity profile I showed you before. Oops, what have I done now? Sorry. I'm getting all tangled up. Okay, it's the same acuity profile I've shown you before for just simple grading acuity from infant monkeys. And on the right, reflected to the right ordinate, is contour integration data. And again, plotted as a function of age in weeks. And some of the data from the same monkeys who we studied for contour integration are also in here. And you can see that these developmental profiles are completely different from one another. And the important thing to notice is that in order to be able to resolve the elements in the contour integration display, you need to be about 3 weeks old. So 3 to 4 week olds could detect the elements themselves. However, they could not do the contour integration task. And that's the point. They could not do this task until they were at least 15 to 20 weeks old, which tells you that it's not about resolving the elements. It's being able to organize that information into some kind of coherent global percept. All right? So there's something different that has to change in the processing of the brain that it certainly happens later than 15 to 20 weeks, that enables us to employ global perceptual skills and to extract that information. So what we're going to do is take a look at 2 different, as I told you, global functions. These data actually are going to be based, one in the ventral stream and one in the dorsal stream. And so most of you know that emanating from V1 and also getting input from elsewhere, just to make sure that we don't leave that out, that there are 2 parallel processing streams through the visual system, the dorsal stream and the ventral stream, and they're largely, they're not completely segregated. There's a lot of interchange between them, but we think about them as being, in the ventral stream, primarily important for processing form information, and in the dorsal stream, primarily for processing motion information. And we're going to start by looking at a dorsal stream area MT, which you've already heard about, and we're going to look at the development of global motion perception and some aspects of the development of NT. So to look at global motion perception, we use stimuli of these kinds, which are random dot kinematograms. And when those dot fields have high coherence, you easily see a coherent image of motion moving horizontally to the left or to the right. And then we can actually degrade that signal by reducing its coherence, and then you would have a much harder time telling what the direction of motion of the overall global aspect of that display is. And we challenged infant monkeys to give us the direction of motion in displays like these as a function of coherence. And so these are data from an individual infant monkey, again, tested at the ages indicated here. And you might now notice that these ages are much older, at the older end, starting at age 2 weeks. And you can see that there is a developmental profile to their ability to do this discrimination task as a function of coherence. These are coherence sensitivities as a function of speed of that display. And you see that again, it's not a consistent ability. It varies as a function of the underlying speed of the dot field. And interestingly, what you find is relative sensitivity is good at fast speeds at the earliest ages, and at slow speeds, largely absent. They couldn't do it at the slow speeds at this early age. As they get older, they are able to cover the full speed range. So this is basically a speed sensitivity function, and it continues to improve up to about 2 years after birth. So an entire year beyond when acuity and some of the other and also temporal sensitivity are developing. That asymptotes by 6 months. So this is actually a higher order process, and it's developing over a much longer time scale. And so we were interested to look at the whole profile of this in a group of animals and then try to relate this to the development of processing in area MT. And over here, what I'm showing you is 2 aspects that change, both the peak speed that I mentioned and also sensitivity to coherence as a function of age for a whole group of infant monkeys. And again, you see these developmental profiles that continue over a long period of time. So what happens at the level of MT? So a number of things change as a function of age. These are data recorded from neurons in infant monkey MT as a function of age at the ages indicated here, 1 week, 2 weeks, and I think 12 weeks, and then adults. And what I'm showing you here is that response measure that I mentioned earlier. In the red here, I'm showing you the development of responsiveness of neurons at the level of V1. And then in green, that same measure at the level of MT. And those 2 functions even are different. So while responsivity changes really across the infant brain as a function of age, it's not necessarily changing in parallel across all areas. And even by age 12 weeks in this series, that was not completely adult-like. And this is another measure that I didn't mention earlier, but is something that also is sort of ubiquitous in the infant brain, and that is long latencies to fire. And this is showing you the same 2 comparisons between V1 and MT, where latency improves in V1 as a function of age, and also in MT as a function of age, but again, much more dramatically at the level of MT. So you have much longer latencies in infant MT. So neurons are waiting a long time to fire. And again, that is not quite adult-like, even at 3 months in this series. So what about direction selectivity? So I told you that direction selectivity in V1 largely doesn't change very much with age. These are polar plots showing direction selectivity in individual cells from 1-week-olds, and this is a V1 cell, and over here is an MT cell. And that means that even in 1-week-olds, neurons in MT, which in adults are more than 90% direction selective. They are in infants as well. And this shows direction selectivity as a function of age in that series, and you'll see it largely does not change. Now, one thing that does change is a property that I think Tony mentioned briefly, which emerges at the level of MT and is not seen earlier than MT, and that's pattern direction selectivity. And to give you a little intuition, pattern direction selectivity, these are just single gratings drifting in different directions, and you combine two gratings on top of one another, and you should get the image of a coherent plaid moving in a direction that's different from either of its component inputs. So you have two gratings drifting in different directions. In combination, they form a plaid pattern that drifts in a completely different direction. And that pattern motion metric that I showed you earlier shows you the development of pattern motion sensitivity. It seems to come in late and develop relatively quickly. And pattern direction selective cells are ones that appear in MT in adults at the level of about 30% of the cells. And we found there were the occasional pattern direction selective cell in the youngest infants, but actually we found one. So we couldn't rule out that they existed, but there were very few. And what you can see is even at 12 weeks, you have a relatively low proportion of pattern direction selective cells. And so that suggests to us that, in fact, there may be a limiting factor at the level of MT when it comes to this kind of global motion sensitivity. And that doesn't apply to basic direction selectivity, which is largely adult-like at very young ages. And so what I've told you here is that properties like direction selectivity that exist in V1, that may be inherited from V1, are already adult-like in newborn MT. But other properties that may subserve more global functions, like those I showed you that develop late or more slowly, and they emerge at the level of MT, and they develop later, even in terms of the physiology. So now I'm going to move on to an example in the ventral stream. And for that, I'm going to take advantage of the fact that Tony just talked about 5 minutes. Okay, that's interesting. That just talked about texture selectivity. What we want to do is say, is there an emergent property, if you will, that's analogous in the ventral stream that will tell us something about global form development? And so what we did was use the same paradigm that he already told you about, which is to look at the development of sensitivity to natural image statistics. And I'll skip a lot of the background. He already showed you that neurons in area V2 will discriminate the natural image statistics in comparison to first-order statistics, but neurons in V1 do not, and that's the profile that we're going to look for in infant V2. Okay, and we used the same paradigm, the Portia-Simoncelli model, where we generated the noise images that have the first-order statistics and the higher-order statistics in the naturalistic texture images. And what we did was, using a variety of families of textures, we generated a behavioral paradigm, a psychophysical task. We tasked animals to tell us, to discriminate which one of a pair was the naturalistic texture as a function of the amount of that natural statistics in it. So we degrade the patterns down to their noise level, and we ask the animals to generate data telling us which one is the naturalistic texture, and we get their sensitivity to the image statistics using a paradigm like that. And so what I'm showing you here is naturalness sensitivity as a function of age for two texture families. Those are the different colors. And these are cross-sectional data from a number of different infants. And you can see that there's a developmental profile. Like pattern motion sensitivity, it develops late Infants younger than this age were not able to do the discrimination, and this 24-week-old here failed at the red task, was able to do the green task, and so there is a developmental onset that's late, like in contour integration, and there is some developmental profile that we haven't fully laid out at this point, but it clearly parallels something like contour integration. So then what we did was we wanted to look in area V2, and we've also looked downstream in V4 and IT, but I'm not going to talk about that. And what we've done is we've used these multi-electrode arrays called Utah arrays. They're 96 electrodes. We've implanted them in particular areas of the infant brain, and I'm going to show you data from V2, and we've recorded from those areas using the exact same stimuli that I just showed you and those that Tony talked about. The youngest animal to date was 26 weeks, another animal at 1 year, and another animal at 2 years. And these are the post-stimulus time histograms from each of those animals, youngest to oldest, from top to bottom. And the same representation here, where the pale band is the response of the population to noise stimuli, and the dark band is the response of the population to the texture stimuli. And you see that in V2, even of the youngest infants, there is some segregation between these two things, but I'll also point out that the scales are different here. Firing rate is much higher, and the response is earlier and much stronger in the oldest, the oldest animal, the 2-year-old, compared to the 2 younger ages. And these are the histograms that go with those recordings. And you can see that there are neurons, even at the youngest age, the 6-month-olds, that will perform the discrimination reasonably well. And at 1 year, there's not much difference between the 6-month-old and the 1-year-old. However, compared to the adult, their responses are much weaker. The discrimination is much weaker. And so we conclude from that that there is some development that takes place. This organization exists in V2 in 6-month-old infants. We have not gone earlier. But we're still working on that, but there's definitely development there, and so there may be another profile. So the take-home from that is that aspects of global perception that rely on extrastriate areas develop more slowly. There's a hierarchical organization, and there are neural correlates of those that may well be setting a limit on those developmental processes. And so I will skip up the rest of it, which was to talk about experience dependence, and I will just tell you that these things do depend on visual experience, and I will thank everyone. So here's my acknowledgement. Thank you, and thank you. My longtime collaborator, Tony Mofshen, other faculty collaborators, and a number of graduate students and postdocs that were involved in this work, and also undergraduates. Thank you.
Thank you very much.
Okay.
I think we have a little time for some questions or comments.
Yeah, sorry,
Please.
I didn't see the clock, so I didn't know how I was doing.
There are several. Okay, go ahead. Professor Wandel.
Thank you, Brian. Thank you, Lynn. Yeah, I'll keep talking and something will happen. I wanted to ask you, kind of back to this earlier discussion from Samir and Tony and now your work. There's a theory floating around that some of the connections, say that Samir was emphasizing, are particularly important during development, that when the monkey's learning, the pulvinar and older pathways that come in are there and present for training, and that as the animal matures, or as we mature, that these become actually smaller in number of synapses and less influential. I wonder if in your experiments and looking at all these many different phenomena of neural development, whether you've seen any hint of these other pathways playing a larger role at 2, 4, 8 weeks, and then by the time they're an adult, maybe their role is less significant, just in terms of that general theory.
There are hints in the literature. There really no good data. We've never done an experiment of the kind that would be required, like to knock out the pulvinar inputs. For example, to MT. One thing that I found interesting that I just learned listening to Samir was this distinction that may exist between the fast inputs, the fast sensitivity from pulvinar pathway, for example, versus slower or maybe a broadband input from V1. And certainly we see perfectly good fast direction selectivity in V1, so it certainly isn't exclusively the case. However, if you noticed in the behavioral data, there is this early emergence of the first things you see is sensitivity to fast speeds. That may be a reflection of a more dominant input at that earlier age, coming from a subcortical input. And take more time for the feed forward from V1 to really have its impact. And, perhaps that's consistent, I'm making this up, perhaps that's consistent with the later development of the emergent properties in MT. Otherwise, I haven't seen, anything that would lead me to believe that. Older in the literature, and actually, Klaus-Peter Hoffmann has some relevant information about this in terms of ocular motor system development, that you might see a transition between a much more subcortical driving to the ocular motor system at very young ages compared to at older ages. And also Claudia Dissler has some information on this. And so there's hints out there that there may be some longitudinal transition in terms of those subcortical influences, where the cortical influences kind of take over. But again, we haven't ruled out that they're consistently there and having an impact.
Thank you very much. Lynne, several more comments and questions. Please, short comments and short answers.
Short answer, okay. I'll try.
Short comment following Brian. The V5 is myelinated at birth, but not fully myelinated. It is one of the intermediate areas that Flexic described, and it gets myelinated as the animal develops. And I wonder whether there's any data about the progression of myelination in V5 and how much of that depends upon input from V1 and how much from elsewhere. But certainly, Flexic's map is very, very interesting in that it is a partly myelinated area. It's not part of the secondary
Beautifully
cortex.
myelinated. Yes, it's a beautifully myelinated area. And there's definitely a developmental profile to myelination that, if I recall correctly, in the macaque monkey covers over at least the first 6 months after birth. But the interesting thing that may be relevant about the latency distributions that we see at 1 week is that we see some very fast ones. So we do see latencies on the order of 80 milliseconds, but we also see a lot of latencies around 200 in longer milliseconds. And so the distribution of latencies is wide, unlike in the adult where it's centered beautifully and very narrow, right around 50 to 80 milliseconds.
Isn't the increase of spatial resolution with age just a matter of an increasing eye size, whereas temporal resolution should be independent of eye size?
Yes, that's a good. He's asking about the changes of acuity as it relates and might drive the development of changes in eye size, and we have looked at that. So the monkey eye changes both in terms of size and in terms of its refractive power, depth of focus, and things like that. All of those anatomical parameters change pretty dramatically as a function of age, but they're adult-like by 12 weeks. And so when you take that into consideration we generate an ideal observer model of the development of the visual system, taking account of that anatomical and changes in the cone spacing as well. And what we found was that at every age, that ability far exceeded the acuity of the animal. And so they didn't set a limit. So while they change, they don't affect the acuity development that we can tell.
Please go ahead.
Thank you. So you showed that the activity of B1 for direction and motion selectivity between the animals in the first week and the fourth week are quite similar. But if you look at it at the behavioral level, then you have a complete different performance. So my question is more about how can we differentiate between the understanding of the animal about the task and the actual ability of the animal to differentiate between the shapes? Because still the activity is similar between these two times.
Two quick things. So first of all, direction selectivity, if you test the animals on direction selectivity, they're basically perfect, just like the neurons are. So this is about the global global motion perception, right? And that's a different task. Second point is that because we generate full psychometric functions for each of these tasks, what we know is that they're able to perform the task at 100% correct if you give them enough coherence. And so what you're seeing is a psychometric function that is steep and well-defined, even at the youngest ages, 2, 3, 4 weeks, and that function shifts as a function of age. So it's not the case that they're compromised in terms of their ability to understand the task or in their ability to perform the task and generate the motor output. Yeah.
Hi. You mentioned at the beginning that humans develop about 4 times more slowly than macaques. Why do you think that is? Do you think there's just no evolutionary pressure or for humans to develop faster, or is there some more profound reason?
The off-the-cuff answer is that they develop 4 times faster, largely in terms of their overall lifespan as well. So if you think about a lot of different species and their developmental profiles, I know I'm very familiar with vision, but there are lots of other things that you could look at. They seem to be scaled often to their lifespan.
2 more short questions back there.
Ursula Pasterkiewicz, University of Waterloo, Canada. Thank you for the fascinating presentation. I found it really very applicable to my research. We currently deal with the devastating influence of very early usage of cell phones. And I'm wondering, I'm struck by one of your final remarks that machinery is surprisingly mature in infants, but the responsiveness is still weak. I don't know if you deal with this in your monkeys, but like high resolution versus low resolution and high speed of motion seems to have a very negative neuronal response. So can you make a little comment on this, please? And thank you.
Please, a short comment.
It'll be hard. I'm not sure I understand the question. So you're talking about high speeds being present early or sensitivity to high speeds and coarse spatial scale.
Let's dedicate this question to the lunch break.
Okay.
Okay. Last question. Please.
Let me.
Oh, you have the...
Hello.
Well, then we have 2 short remarks. Please,
Sorry.
go ahead. It's a very interesting talk you see.
Thank you.
Adult animals will take a little bit of time to become experts in a task. So have you tried to compare the rate of learning, the rate of learning of an adult versus a macaque monkey? Because then this might change a little bit, the picture you presented. Maybe they become mature earlier in a way in doing some tasks. Is that...
Oh, yes. So do you mean that so our developmental profiles may be contaminated by the fact that there are actually expert psychophysical subjects as infants? Is that the question? So I'll just say that could be the case, but what we do is we also include animals that are tested cross-sectionally only, so they haven't had that early experience, and they learn at about the same rate, and their sensitivities are similar. It's a great question. It's a great question.
Just about the learning process of the global properties. Is in your data is more related to, or could be more related to, more basic mechanisms like Hebbian learning in a bottom-up manner, or maybe inferential processes like the children has to do some sort of modeling of the world. Then, so do you think that learning will be more related to more basic bottom-up or top-down processes, or both? The learning of the global properties will be more related to very basic processes like Hebbian learning, or maybe more complex processes like inferential models?
And I think
I think there's a lot of Hebbian processing that's going on very early in the infant brain at the synaptic level. But I think there's structural changes that happen in these areas that underlie the global form and motion.
Thank you very much.